Very good afternoon, dear students. We have been discussing various applications of environmental microbiology in the open course and interdisciplinary science. And first three units we have discussed about biogeochemical cycles and the soil system and some aspects in the water and aquatic ecosystem. And the next important two units we are going to focus how the management of microorganisms and waste we are going to carry out. Okay, and in this particular unit, module number 4, this is mainly deals with the dispersal of airborne microorganism. So in the previous class we have discussed the microorganism that is present in the aquatic ecosystem, and in this particular unit we are going to see what are the major microorganisms present in the air and what are the airborne microbial flora and how are you sanitize the air and purify the air and how are you going to disperse the microorganism present in the air. So as an introduction, how the air act as a source for the growth of microorganism. So the question is an air, okay, air is a good source of a medium for the growth of microorganism? The answer is no, okay, because air, it is not a natural medium for the microbial growth and their reproduction, because no significant nutrients that is required for the growth of microorganism and moisture content present in the air, okay, and air, you know that doesn't have nutrients and that doesn't have so much of moisture in all the season, okay, and which undergoes so much of environmental fluctuation. So the study of living microorganism which are suspended or distributed in the air system is known as aeromicrobiology. So aeromicrobiology is the branch of environmental microbiology which deals with the study of living microbes organism which are suspended in the air, okay, and the microorganism that is mainly introduced in the air by several means, okay. The principal source of microorganism in the air are the dust particle, okay. The air has lot of dust particle and the dust particle will provide the suitable surface for the growth of microorganism, okay. So this dust particle contain both the normal cell that is known as vegetative mature cell and the resistant cell that is a dormant cell that is known as spore, okay. So the dust particle contain both the normal cell and the unusual that is resistant form, okay, and some are saprophytes which are originated from the dead organic matter. You know that the microorganism act as the decompose when the bodies of animals, plants, microorganism, okay, all those organism when it is died, that will be reached into the soil where the microorganism degrade that particular dead material and that will be contributed to the soil and the microorganism can introduced in the air, okay, and the number and the variety of microorganism present in the air are based on the locality, okay, which location the air is situated. Based on that one, the number and kind of various microorganisms are present, okay. Generally the fungi are most predominant than bacteria, okay. Fungi, you know that fungus classified into two groups, one is mold, another one is yeast, okay. Both this mold and yeast are predominant than the bacteria, okay. Then how the microorganisms are distributed in the air, okay. The number of microorganisms in the air primarily depending on the activity activity of the environment and the amount of dust particle. I told you the dust particle is the major source of substrate that is going to present in the air, okay, and the activity of environment, okay, what are the activities are going into that particular environment, and what is the amount of dust particle will be primary reason for the presence of microorganism in the air, okay. An active environment shows higher bacterial count than the less active environment, okay. An environment is highly active with a lot of activities, okay, are going on. That environment has higher number of bacteria than the an environment that is very less active, okay. So definitely a dirty, untidy rooms have more number of microorganisms than a clean room, okay. A clean room doesn't have that many microorganisms, but a dirty room or untidy rooms, okay, has a lot of unorganized surfaces that has so many microorganisms. Similarly, the tropical air consists of large number of gram-negative bacteria than and gram-positive pleomorphic bacteria than other type of bacteria, okay. A tropical air consists of higher percentage of the bacteria, you know that classified into two groups based on the cell wall composition. What is? One is gram-negative bacteria, they are rod shaped, and gram-positive bacteria. Pleomorphic means the bacteria doesn't have exact perfect rod shaped bacteria, they are able to change a little bit, that is known as pleomorphic, okay. Then these two bacteria are predominant than other type of organism. Then if you take the soil, that is the air that is associated with the soil, okay, that means a rich, fertile, cultivated, fertile soil contain large number of microorganisms than the sandy or clay or uncultivated soil. In cultivated soil, you know that lot of vegetation will be there. The soil, the application of fertilizer, lot of vegetations, lot of animals, plants, so many things are there. Due to the presence of all those things, lot of microorganisms are present on the surface of all these organisms. So that will simply distributed in the air, okay. Similarly, the terrestrial air contain more organisms than the marine air, okay. If you look at the marine ecosystem very near to the ocean, that particular environment has a less number of organisms than a tropical or a forest or other greeny or terrestrial air, okay. Then similarly, a humid atmosphere or damp atmosphere contain more number of organisms than the dried environment, okay. If you are having a dried, highly dried sunlight, okay, that environment has a more less organism than the humid environment. The humid environment, there will be particles, so the microorganism will be present there. Similarly, inside the refrigerator, air conditioner, AC room has a less number of organisms than a normal room. Refrigerated air generally free from organism, or in case there are some of the organism will be there, they are very lesser than the other environment, okay. And the seasonable variation also has lot of importance. Dry summer season contain more organism than the winter season, okay. The summer season has more number of organism than the winter season. You know that the reason winter season always rain, the temperature is very lesser. Similarly, but in the case of summer season, the temperature is always there, that is the 37 degrees centigrade is helpful for the growth of bacteria. So that is why summer season is has more number of organism than winter season, okay. Then winter season temperature is very less also. Then a lower altitude has high number of organisms than the higher altitude, okay. When you are traveling from the earth surface towards the top, okay, the region in which the air in which that is surrounded by the soil, that has a more number of organisms than when you are traveling from towards the higher altitude, okay. So towards near sun, the microorganism microbial count will be very less because temperature is increases, okay. But in the lower moreover, there are a lot of microorganisms that is arises from the all the soil, in the animals or vegetables and vegetation, etc. Okay, so these are the microbial distribution of the air. And what are the major parameters that influences the presence of microorganism in the air? There are so many parameters that mainly influence the growth of microorganism in air. So for example, the speed of air current. Okay, when you are speed of air current means the wind. Okay, when the wind is circulating, the microbial flora will be distributed throughout the air. Okay, similarly, period of time. Okay, and what is the duration of time, the size of dust particle. If the dust particles are in a very higher size, there will be larger number of microbial particles will be attached to the dust particle. Then the humidity of that atmosphere. If the atmosphere is humid, I told you the number of microorganisms will be very high. Okay, then density of human and other animal population. Okay, the population of living organism is high due to the activity of lot of living organisms. So many microorganisms are expelled to the atmosphere. Then the nature of the soil. I told you the fertile soil has a lot of bacteria and other microorganisms than a non-fertile soil. Okay, then amount of vegetation. Okay, if the plants, the green plants and vegetations are more, the microorganisms are also more. Okay, and the temperature. I told you the temperature. Summer season has lot of microorganisms than the winter season, because the summer season they have a lot of adequate temperature than the winter season. This I have already told you. The wind circulation, the flow of wind from one direction to another direction, that is spread the microorganism everywhere. Okay, rainfall and sunlight. Okay, rainfall, when the rain is falling, the microorganism will be very less, but the sunlight, the microorganism will be more due to the variation in the temperature. Then these are the some of the activity you can see that contribute to the microbial source to the air. When people are coughing, sneezing, the lot of vegetation. Okay, then the agricultural practices, when you are mixing the soil with the fertilizers. Okay, then dumping the waste in the water body, then the activity of various various organisms. Okay, such as domestic animals. Okay, similarly human, the industrialization. Okay, then there are lot of microbial flora will be emerged from this particular activities. Okay, then here you can see this is electron micrographic photograph of the when you are coughing, sneezing. This is very very important at this present, this COVID-19 era. You can see when a person is sneezing or coughing, how many droplet fine particles are generating through the atmosphere. You can see, and this has lot of water droplet content along with the microorganism. That is why we are telling you we should always wear mouth mask to prevent this COVID-19. Okay, so that is the most important thing. This is the clearly indicating how the microorganisms are expelling to this particular air. Okay, whenever you talk, cough, sneeze, this is what is happening. So a person who is standing very near, it will be coming, the microorganism, especially the viruses and other things, will be coming from one particular host to the another particular host. This is another important source of microorganism. Then what are the major microbial flora present in the air? What are the various kinds of microbial flora present in the air? So one is always in the free form, that is known as vegetative form. Okay, so whenever adequate temperature, nutrients are available, they are able to survive in the normal temperature, normal form, normal shape, that is known as vegetative form. Whenever there is a lack of proper nutrition or scarcity of the food, the microorganism, they are able to resist that condition by changing the morphological form called endosperm. If it is a bacteria, that is endospore. Okay, then it is fungi, that is exospore. Okay, then these are the very very important. I think this will be you will be heard during especially this COVID-19 pandemic. Okay, aerosols, or droplet nuclei infectious dust. Okay, so these are the various kinds of microbial flora. And what are aerosols? So I have already told you first two. The third one is, this is a biological contaminant simply suspended in the air. So it may have the size of 1 micrometer to 50 micrometer in size. And what is this particle is about? This particle composed of the microorganism attached to a dust particle. Okay, the microorganism attached to any type of dust particle. There are two types of aerosol, and this biological origin is known as bioaerosol. Okay, one is the bioaerosol generated from human sources, I told you, okay, and bioaerosol generated from environmental sources. Whenever you cough or sneezing, lot of small particle that is with the water droplets along with the microorganisms are arises. That is known as bioaerosol. Next is, this is very important, droplets. Okay, this droplets are one of the major transmission of COVID-19. These are the small droplets of water or moisture that combines with the microorganism to form a solid residue. How it will be producing a solid residue? Okay, so what will happen? This airborne droplet nuclei develop when the fluid pathogenic droplet, that means to say fluid pathogenic droplet in the sense the water or moisture combines with the microorganism, and these droplets are smaller than that of the aerosol, that is 1 to 5 micrometer in diameter, that produces a solid residue and which will evaporate from this particular residue. Okay, then it produces a small residue called droplet nuclei. So the droplet nuclei contain pathogenic microorganism, and this can survive air for very long time and they can cause the transmission. If you look at the aerosols and droplets, they can transfer up to 6 feet. That is why the social distancing is very important. Okay, so that is the major reason why social distancing is important. Next one is infectious dust. The dust particle combines with the pathogenic microorganism that may arises from human or environmental sources. Okay, the dust which are arises from human or environmental sources, that is known as infectious dust. The dust generated from either from human source, human activity, whenever coughing, sneezing, talking, etc., or environmental sources. Okay, from animal, from plants or vegetation activities. Okay, all those kind of activities. So these are infectious dust. And due to all those, organism, they cause us airborne infections. Okay, what are airborne infection? The infection caused via air by various microorganisms, that is known as airborne infection. They are mainly airborne bacterial infection, airborne viral infection, airborne fungal infection, airborne protozoan infection. Okay, so some of the examples: airborne bacterial infection, tuberculosis, that is caused by Mycobacterium tuberculosis. Okay, a bacteria that is that is a lung infection transmitted through air. Okay, next one, diphtheria. Okay, causative organism is Corynebacterium diphtheria, that is also transmitted through air. Okay, the next one is meningococcal meningitis. Okay, the organism that is caused, that is meningitis, is Neisseria meningitidis, that is also transmitted through air. Okay, then you know the airborne viral infection, you know that COVID-19, that is SARS coronavirus 2, common cold, okay, influenza, smallpox, and chickenpox. These are the airborne viral infections. Okay, then the next one, airborne fungal infection, mycosis. Myco means that is fungus. Any type of fungal infection transmitted through air is known as mycosis. Histoplasmosis, that is caused by a fungi called Histoplasma capsulatum. Okay, the next one is cryptococcosis. Okay, then this particular infection is caused via air by a fungal yeast called Cryptococcus neoformans. Okay, then airborne protozoal infection, blight grain disease, and protozoan pneumonia. Okay, so these are the various types of airborne infection. There are many more. Since you are this just an introduction, only this much things you should understand. Then why we require the dispersal of this microorganism from the air? What is the need of sampling of air? How will you sample the microorganism from air. Okay, so this microbiological air sampling is very very important to purify the air in a particular environment. It is used to determine the number and types of microorganisms in the indoor and outdoor air. Inside the room and outside the room, it is very essential to study the number of microorganisms. Then the air sampling for the quality control is a problematic because there is a lack of uniform air quality standard. This is doing, even though this is an important method, okay, but we don't have that much of good quality control, good standard to compare the result what we are going to get. So thus this is a very tedious process. The indoor air quality may affect based on the following parameter. What do you mean by indoor air? The air that is present inside a room is known as indoor air. Okay, indoor traffic, okay, that can be human traffic, okay, animal traffic, okay, or wind traffic, okay, visitors entering facility, visitors are going out and in, temperature inside the room, the time of the day or year, how much time we are spending, many meetings are happening inside the room, then the relative humidity, the concentration of particles and organism, performance of air handling system and components, okay, the AC, similarly fan, okay, all those things will spread the microorganism inside a room, okay. So that is very essential to understand the microbial quality that is present inside the air, okay. So then what is the need of air sampling or dispersal of microorganism? Number one, to protect a product integrity, food and beverages, because food and all those things we have kept inside a room. If the air is not good, the microorganism will come and fall into the food item. Then the prevention of hazardous microbe present in the atmosphere. We need to prevent the microorganism present in the environment. Okay, so you know that major the route like COVID-19 is transmitted through air. Okay, then improved quality and increased safety to maintain the quality and the to maintain the safety. Okay, next one is the reduction of overall production risk and expenses. Okay, reduction of overall production whenever you are producing something, and to control the risk and to expenses. Okay, if air contaminants are more, the processing of that particular items will be very high to remove those microorganisms. That is the reducing the risk and expenses. Then increase the quality of control, okay, to maintain the healthy life, public health, safety and hygienic ecosystem, and that is very very important. These are the main objectives of the sampling or dispersal of microorganism present in the air. And what are the major methods for the sampling of air and dispersing the microorganism or collecting the microorganism from the air? There are mainly 7 different principles. One is known as impingement in liquids. Next one is known as impaction on solid surfaces. Next one is known as sedimentation. Next one is known as filtration. Next one is centrifugation. Next one is known as electrostatic precipitation. Next one is thermal precipitation. So these are the various methods for the sampling of air and dispersal of the microorganism in a particular air. Now we will see very very briefly topic is one big unit in microbiology and bacteriology, but we will discuss only brief concept as you are studying very basics of this particular topics. Okay, first one, impingement in liquid, and the main method impingement in liquid. And what is the principle? We will see that what is the method, what is the principle, and what is this particular method is useful, and what is the main microbiological parameters are using, and what is the rate of collection or dispersing the organism, and what are the main instruments. Okay, so these are the things we are going to discuss. Then the method is impingement in liquid. It's very simple. The air drawn through small jet and directed against a liquid surface. Okay, so just you in a very small minute tube collect the air, and one end just you dip into a liquid surface. So in a particular time you expose, take that particular medium and incubate. Next day you will be getting the bacterial, incubate and plate into a medium, you will be getting microorganism. So it is suitable to measuring all the living organism. Okay, from what are this liquid medium, liquid surface? That can be buffered gelatin or saline or peptone or any other broth, or even your food. Get some food and make a suspension. That one also can be used. The rate of collection is 12.5 liter per minute. An example is a lemon sampler, all impinger, that is known as all gas impinger AGI. Okay, that is the one example is lemon sampler, that is known as impingement. Impingement means just to impinge the air to a small tube and collect into a liquid broth. Next one is impaction on solid surface. So the method is impaction. So same way the air is drawn into the sampler and the particle deposited on a dry surface. Collect the air and allow the air to deposit into a surface. That surface has a medium. The microorganism present in that particular air will go and deposit, and that surface you incubate. Next day you will be getting all the possible number of bacteria that is present in that particular air. Next one is viable particles, that can be used to count the viable particles, the viable organism. Then size also you can measure. Okay, and any dry surface or coated surface or agar surface, a medium, okay, we can use as a surface. Then this is the rate of collection 28 sieve or 30 to 800 slit, sieve and slit. Physics students will be knowing. It's a very common you things that whatever you are using for experiments. Then the prototypes are samplers are Anderson air sampler and a capillary impinger. Both the things are working based on the impaction on solid surface. Next one is sedimentation. You have already studied what you mean by sedimentation, that is settling. Okay, sedimentation is the method and principle. The particles and microorganisms are settled at the bottom due to acceleration, due to gravity. Okay, so you know that whenever you keep some air, that air particle will be settled at the bottom due to the gravity to any surface. Then that surface you take and identify the number of microorganisms present in that particular surface. So here, which is suitable to study all the viable bacteria or viable particle. So what are these surfaces? That can be a nutrient agar or any glass slide or plates which can be used. And there is no specific rate, and any type of settling plate can be used as a sampler. Okay, that is all about sedimentation. Next one is filtration. The name itself, it is there. You filter the air through a membrane. That's all. Okay. So filtration, the air drawn through a filter unit, particle trapped 0.1 micrometer pore size. We have already discussed membrane filter, isn't it? So just you pass the air through a membrane filter. This membrane filter has a lot of tiny holes, that through that holes the air will be passed, but the microorganism will be trapped. That trapped microorganism just you transferred to a microbiology medium and you incubate. Next day you will be getting the growth of bacteria. Then that is suitable for both the viable particles and viable bacterial count. There we have lot of example: paper, cellulose, glass wool, gelatin and membrane filters as the surface. Then the rate is generally 1 to 15 liter per minute. Okay, the prototypes, there is no prototypes are applicable. Next one is centrifugation. This you have already studied. Okay, centrifugation. So the particle like aerosol subjected to a centrifugal force, the particle will be impacted on the solid surface. What will happen when you apply a centrifugal force? Microorganisms are small particles. Okay, with respect to the centrifugal force, the microorganism will be segregated or dispersed and settled. Okay, that is known as centrifugation. Then the microorganism will be settled at the bottom. Okay, then the suitable medium that is suitable for viable bacterial count, and the media can be used. The coated glass or plastic slide, okay, or agar surface can be used as a solid surface. Here the rate is little bit high, 40 to 50 liter per minute. The rate of collection can be done. Then this is a standard biotest RCS plus. This is a typical centrifugation instrument made by a company. Okay, so that is another example. Next one is electrostatic precipitation. So electrostatic precipitation. You know that when you are talking about electrostatic precipitation, there will be both the positively charged electrode and negatively charged electrode, both anode and cathode. You have already studied. Okay, so electrostatic precipitation, what you need to do is the air drawn over an electrostatically charged surface, okay, and the particle become charged. Okay, so what will happen when the air is passed through electrostatically affected region? Whenever you know that microorganism has, most of the microorganism has negatively charged surface because of the presence of certain macromolecules, and there will be deposit towards the positively charged electrode. Okay, so that is, then it will be settled at the near the positively charged electrode. Then that can be used to study the viable particles, okay, and organism. The solid collecting surfaces such as glass and agar is the example. So the rate is very high here, that is 85 liter per minute can be done. The prototype sample is that is not applicable. So electrostatic precipitation, here the air is passed through electrically charged an electric charged place where the both the positive and negative charged electrodes are there. Okay, then it will be separated based on the charge. Next one is thermal precipitation. Here it will be based on the heat. Okay, so the thermal precipitation, air drawn over thermal gradient, the particle repelled from hot surface settled on the colder surface. Okay, so the particle, the microorganism that always repelled from the hot surface, that moves to the cold surface and settled. Okay, where whatever the microorganism, when you are the air is, whatever the air you wanted to sample, collect the air, that air you are drawn over a thermal gradient, that is subjected to the thermal gradient, that the microorganism present in the air will be repelled from hot surface. When you are applying for a thermal gradient, the temperature will be increased. Then what will happen? The microorganism will be expelled, repelled from the hot surface, then it will be settled at the cold surface. The size measurement is the best suitable approach to study this one. Okay, then the main collection media, that is glass cover slip, the electron microscope grid. Okay, this is very small. This is the smallest rate of collection, that is 0.0003 to 0.4 liter per minute. Okay, there is no sample, there is no specific sample types. Okay, so that is the prototype samplers. Okay, so these are the common air samplers. This is known as lemon air sampler. Here is there is a medium. Here you can see there is a scale, it is kept. This is a very narrow tube. The air is drawn over here and it is dipped at the medium. Then once you close this one, the air will be passing here and the microorganism will be grown over here. Similarly, this is capillary imbinger. Here you can see a capillary tube. Here you can see a conical flask. There you can see there is a medium. Okay, the air sample will be passed inside. Okay, then when you pass in a specific time, you release this one and the microorganism will be distributed over here, and the next day you will be having the growth in this medium. Then this is known as Anderson air sampler. Here you can see this is in a big chamber. Okay, this is one medium. The second medium, a plate consisting of medium. 1, 2, 3, 4, 5, 6. Like that, one over the other it is kept. And here you can see the their air is traveled from the particular port in a particular time. Then the air will be moving over here. Okay, now all the chamber it will be moving. Okay, so the maximum air will be segregated here. Then it will go there. The microorganism and it will move towards this particular region. The microorganism, you will be getting less number of bacteria in this particular plate. Then comes this one, then comes this one, then comes this one, and then only comes the last one. This particular plate has the maximum number of organisms. So you can count and you can sample the air from all the microorganisms. That's all, dear students. We are going to conclude. Okay, so today we have discussed what is the importance of air microbiology. We have discussed and why air need to be sanitized, air need to be purified, and what are the microbial distribution in the parameters in the air, what are the various factors which influencing the presence of microorganism in air, what are the sources of microorganism in air, what are the major kinds of microbial flora, okay, that is vegetative cell, spores, then aerosols, droplet nuclei, infectious dust, okay, all those things. Then what are airborne infection, airborne bacterial, viral, then fungal and protozoal, okay. Then the microbial sampling of air, what is the necessity and need and objectives of microbial sampling, what are the factors, and what are the major methods of air sampling, impingement in liquid, impaction on solid surface, sedimentation, filtration, centrifugation, electrostatic precipitation, thermal precipitation, etc. So this is all about this particular unit, okay. So these are the main references, okay, as I said. Then thank you very much for listening this particular class, okay. If you are having any clarification, you can ask me. Thank you very much. Have a great day.